regressed as drivers. Like there's construction on the 406 right now. And all you have to do, and we all know there's construction on the 406, it's been there for weeks, right? And all you have to do is you know that heading St. Catharines bound, like you're going to have to merge into the left, right? We all know this. So like, to me, since we all know this, why not just the second you get on, just get to the left and drive? There's plenty of room. All the cars fit in one lane. But they don't. People stay in two lanes. And then right up to the last possible second, people stay in and then try to get over at the last second. And then because the people in the left lanes are also jerks who don't know how to drive, they don't let people in, so then everyone has to jam on their brakes. And that turns down into a big traffic jam, despite the fact that one lane is more than enough space to fit all the cars that are going, because people are just so discourteous and stupid that we can't like merge in like a civilized people. And then like I counted, in the last 20 days I've been driving home, 6 times there's been a car accident where that merge in is. 6 out of 20. I mean, that's an astounding number, right? Like it's depressing, really. Can I tell you quickly, driving lesson? This is the only thing they should teach. You will never get in a car accident ever in your entire life that is your fault if you do 2 things. 1, obviously, pay attention to your surroundings, look before you change lanes, know where you're going, act appropriately. Don't move your car sideways until you're very confident that on either side of you there is in fact an empty space to go into. And 2, leave enough space in front of you that you have time to brake if the car in front of you comes to an immediate stop. And you have to factor in an immediate stop because they may hit the car in front of them, which means they can stop faster than the brakes could stop them. So you have to make sure you're back far enough that when they stop immediately, you can come to a stop, not immediately, because immediate stops mean you hit something. Does that make sense? As long as you do that, you will never get in a car accident as your fault. Simple as that. And then the second thing is, if you want to be a civilized driver, where do you drive? Yes, to the right. You just drive to the right, and then you only ever pass somebody on the left. It's simple as that. You drive on the right, and then when the person in front of you isn't going fast enough for you, you go to the right, you go in front of them, and then you get back in front of them, and you keep driving. And then you should never be in a lane other than the far right lane and have somebody up behind you. Because if you're in a left lane and somebody's up behind you and you're going, oh, look at this idiot tailgating me, just get over. Just get over. If somebody's trying to go faster than you, you should let them, and you should let them pass you on your left by getting to the right. Because that's what you do. You pass people on the left, but you get these people who are like, I'm going to drive 105 in the left lane, and I'm going to be pissed off that people are tailgating. Just get over. What's the big deal? Leave lots of room in front of you, get over, and then anytime somebody tries to get in, whether you like them, whether you hate them, whether they're jerks, whether you think they tried to cheat and get right to the end, if somebody tries to get in front of you, let them in. By the way, if you're leaving enough space for safety, you won't have to let anyone in because there should always be enough space in front of you for a few cars to fit. Because if you're so close to the car in front of you on the highway that nobody can fit between you, you're not driving safely. And if that car in front of you comes to a quick stop, you have no chance of stopping yourself. All right? So you should never even have to let anybody in. There should always be room in front of you for a car to get in. And as long as everyone does that, there'd be no traffic jams. But anyway, this is why we can't have nice things. Let's talk about Krebs cycle. All right, when last we met, we took some glucose, we underwent the process of glycolysis. From that, this says 2 ATPs. I thought we got 4. Why does it say 2? Nope, I like that thinking. Because we had to use 2. So this is just showing the net. Does that make sense? How many ATPs do we actually get? We actually get 4, but we have to pay 2. So in total, we get 2. It shows 2 NADH from glycolysis. Yep, absolutely. Those NADH from glycolysis are going to move into the mitochondrion. And then, as the pyruvate, which you'll also see called pyruvic acid, that's the same thing, it means the same thing. As the pyruvic acid moves into the mitochondrion, it gets processed into 2 acetyl groups. And when they actually move in, they're bound to what? This thing called coenzyme A, so we call it acetyl-CoA. But the truth is, the acetyl group is actually the 2-carbon sugar, or the 2-carbon molecule left over from from pyruvate. Now, pyruvate has how many carbons? 3. So what had to get released between pyruvate and acetyl coenzyme A? Yeah, we released 2 CO2s. 2 of them because we had 2 pyruvates, right? So each pyruvate gives up a CO2 to form an acetyl group, which is bound to the enzyme coenzyme A. So 2 acetyl coenzyme A's move into the mitochondria. Yeah. It's literally while entering the mitochondria. I don't love this diagram because it really is while entering. So it's literally the transport protein in the membrane of the mitochondrion is coupled with the enzymes responsible for making this reaction happen. So it happens while entering the mitochondria. And that's a good question, by the way. Okay, so now what do we do with the rest of the energy? We still have a bunch of energy stored inside of this acetyl group. Yeah. Sorry? That's an E. That still doesn't look like much of an E, but it is an E, I swear. There you go. Is that better? Yeah.
So the COA, is that just like after it like brings the 2 acetyl? Does it just kind of like chill and like swim around?
Well, we can talk about that. No. So the whole point of that coenzyme A is to make it this big bulky molecule that is going to get attracted to the mechanisms inside the mitochondrion right away. So remember that there's not some crane moving things around. These things have to happen by just sort of bumping into each other and having an affinity for one another, right? Like being attracted to one another and doing things. So the coenzyme A molecule is designed to be highly selective for acetyl groups. In other words, it sees an acetyl group and boom, it binds to it and it's highly attracted to acetyl groups, so it will find them. Does that make sense? Once it has done so, the acetyl coenzyme A, the coenzyme A is also designed to be highly attracted to the mechanisms that dictate the next thing we're going to see, which is what we call the citric acid cycle or Krebs cycle. Okay? And Krebs cycle is basically the factory floor where processing sugars starts to happen at a clip and to be highly effective. Does that make sense? Glycolysis is a fairly inefficient process. It takes 10 steps to get 2 ATPs and 2 NADHs. You're going to see now in the mitochondrion, which is, of course, as you'll remember, powerhouse of the cell, you're going to see that there's a process designed that very quickly gets a lot of energy out of this remaining acetyl group. Does that make sense? We're going to get a bunch of energy out of that acetyl group, and we're going to get it out quickly, but we're also going to get it out in a way that is very ingenious, that's very efficient and clever. Does that make sense? So let's take a look at it. By the way, the citric acid cycle was discovered by a lab group run by Hans Krebs in 1937. This guy's a biochemist from England.
Morning. Pardon the interruption. Could we have Sarah Chow in guidance?
Yeah.
Thank you.
Sarah Chow? Yeah, I heard that one. So we often call the citric acid cycle the Krebs cycle. Note, it's not like his name was Krebs and this is Krebs cycle. Rather, his name was Krebs, and so it's the Krebs cycle. But I more often call it the citric acid cycle. Anyway, let's take a look at what the hell is going on here. So first things first, this takes place in the matrix of the mitochondrion, all right? So in the matrix of the mitochondrion, we have all of these things. And to be clear, these are all just kind of floating around, but here is the basic idea. This acetyl group, and I wish that this kind of highlighted this more fully, this is what remains of glucose, right? And rather than working on that acetyl group by itself, what they do is they tie it to bigger things, because working with a really small molecule has a big disadvantage, which is small molecules move around freely and they float away. Does that make sense? So we stuck it to this acetyl coenzyme A because that makes it big and bulky and easy to control where it goes. Does that make sense? Now, if all we do is just take it off the coenzyme A, we'll be right back at square one, where that acetyl group will want to float around. So instead, this process is designed, look at me, see if we can work around this, to say, what if, instead of processing a small little molecule, which is all that's, and we want that pyruvate to be small to get into the mitochondrion, because that's advantageous for moving it in there. Once it's in the mitochondrion, they say, what if we attached it to a pretty big molecule, and then we processed that all the way through, so that when we're done, we're back with the really big molecule we attached it to, and we can attach another acetyl group to it and work it through again. And then when it's worked all the way through, we're back with the big molecule we started with, and then we can attach another acetyl group to it and have it go through again. But then the whole time we're working with big molecules instead of little delicate ones. This is only an analogy. You can imagine it's a lot easier to do woodworking on a big scale, like framing a house, than it is to do delicate little woodworking that involves really small hand functions. Can you kind of picture that? Working with big things is advantageous. So, what basically happens then is this. This acetyl coenzyme A is going to come in, and we're going to add a water molecule to it, which is interesting, because water mostly gets released by this process. But at this stage, we actually use a water molecule. That's okay, because what are cells full of? Water. So we've got lots. But we're going to apply a water molecule, and we're going to take this molecule called oxaloacetate. Metabolites, we'll get there, don't worry. Metabolites are just the products of metabolism. So we're going to take this oxaloacetate, do we see that? Which is a little bit bigger. How many carbons do we have here? 4. And we're going to stick our acetyl group onto it, right? Which means we'll be back to dealing with how big a molecule? 6 carbons. Does that make sense? And that just makes this thing easier to work with. And what will that product be called? We take the oxaloacetate, and we're going to add our acetyl group to it, forming something called citrate. And look, 1, 2, 3, 4, 5, 6 carbons. Can we see that? And specifically, there is our acetyl group, basically. There's the oxaloacetate molecule, and what have we stuck onto it? A citrate. In fact, we could show all 3. Oxaloacetate and acetyl. We'll call it acid. We'll call it. I got this. Ethanol and citrate. Let's see if it'll give me all 3. Well, that might take all day the way this thing's running today. But while we wait for it, so look here. We take this molecule that's already in the cell. In fact, this is the starting point. This is already in. All right, this is already present. And we add to it our acetyl group. Does that make sense? So that means this coenzyme A just gets released, which is good, because what can that coenzyme A go up and do? Well, it can go back and attach itself to another acetate group. Does that make sense? Or another acetyl group, I should say. I wonder if that's loaded now. It didn't like me. You used to be able to just separate these with and. I wonder if it doesn't let us do that anymore. Anyway, doesn't matter. Okay, now what's going to happen is this bigger molecule, we're going to go through a series of reactions to turn it back into oxaloacetate. Does that make sense? And when we've turned it back into oxaloacetate, that means we will have dealt with every little bit of energy that was in the acetyl group that we added to it. Does that make sense? We're going to take this oxaloacetate, which is sort of like the basic molecule we're starting with. We're going to stick our 2-carbon sugar onto it, and then we're going to slowly modify it so that we end up back at oxaloacetate having taken all the energy out. Does that make sense? We're going to go through step by step, but really quickly, can anyone see in here steps where they can see that energy gets taken away? Where we're going to absorb energy. There's a molecule you should be familiar with that shows up here. In fact, it shows up 3 times. NADH. Do we see that? Here we release an NADH. Here we release an NADH. Here, or I shouldn't say release. We form an NADH. We form an NADH. And we form an NADH. Does that make sense? Well, the citrate, which remember has our acetyl group, which is what's left of our sugar, at this stage, our sugar is going to lose some energy. Now, what gains that energy? This NADH molecule, which we're then going to turn into ATP later. In other words, we're taking the energy out of this acetyl group and we're storing it for our usage in a way that the cell can use. And then at this stage, what happens again? Again, we take some energy out and we store it. What happens at this stage? Same thing. Now, as it happens, we're going to meet 2 other storage molecules here. Another storage molecule, instead of adenosine diphosphate turning into adenosine triphosphate, here we have guanosine diphosphate turning into guanosine triphosphate. By the way, you're used to adenosine, adenine, and guanine, right, being nucleotides. Well, here, just like adenosine can be used with phosphates to store energy, so can guanine. This will actually later just get converted to an ATP. But for now, and the other one you're going to see here is FADH2. All of these are places where energy gets released. Okay? Let's take a look at that. We're going to highlight all these and say, this is where we capture energy. This is another place where we capture energy. This is another place where we capture energy. This is another place where we capture energy. And this is another place where we capture energy. An NADH is good for about 3 ATPs. So this is the equivalent of 3, 6, 9 ATPs per pyruvate. And then a GTP is pretty much equivalent to an ATP, so that's a 10th. And then an FADH2 is good for about 2 ATPs, so that's 12. So through this process, each acetyl group will give us the equivalent of about 12 ATPs worth of energy. But of course, acetyl comes from pyruvate. Pyruvate came from glucose, but glucose didn't give us 1 pyruvate. Glucose gave us 2 pyruvate. So every glucose gives us 2 runs of this cycle. Does that make sense? Which means this cycle for every glucose is good for the equivalent of about 24 ATPs. Compared to glycolysis, those 10 steps only gave us the equivalent of 2, and then plus you get 3 per NADH, so about 6 more for that, so about 8. So the whole process of glycolysis was good for roughly 8 ATPs. This process is good for about 24. Does that make sense? It's substantially better, yeah. That's right. And that's because this coenzyme A gets reused. So the coenzyme A that was used to bring the acetyl group here will go back and it'll bind to another acetyl group and it'll bring it down here. And then it'll get released and it can go up and get another acetyl group. So you don't have to make a new coenzyme A every time a glucose enters the cycle. That would make this process too expensive. Just like you wouldn't build a new shopping cart every time a shopper comes to the grocery store, you just reuse them, right? You get very expensive making bespoke grocery carts for every customer who came to the store. Yeah. The NADH? That's good for about 2. And don't worry, you're gonna learn all that, so you don't have to write that down now. But does that make sense? These are the steps where we capture energy. Okay. What else do we want to notice here? Well, we want to notice that this citrate is basically the oxalatoacetate. Oxalatoacetate, which we more often just call oxaloacetate, which is fine, plus that acetyl group, right? And this is our starting point, right? So if that's the starting point before the acetyl group, now this is like the starting point where this is the starting molecule already in Krebs cycle. And this is what remains of our sugar, right? And how many carbons did we say this was? We said the oxaloacetate was a 4-carbon molecule. And of course, the acetyl group is how many carbons? 2. From the Krebs cycle. Right? In other words, that oxaloacetate is always going to be present, and it's going to get an acetyl group added to it to form this citrate, or this citric acid, which is a 6-carbon sugar. So through the course of this cycle, what are we going to have to lose 2 of? Carbons. Where does that happen? Well, the first thing that happens is just a rearrangement. This thing called aconitase, or aconitase, I've never known how we say it, takes citrate and just changes its shape to form isocitrate, all right? And if MolView is being cooperative, you'll see there that that doesn't have a huge change. So if we go citrate, does it want citric acid? Does it just not want to work? Well, that's fine. So there's citrate. Do we see that? Now if we look at isocitrate, see the difference is pretty minimal. Come on. It's just a rearrangement. I wish I could put the 2 next to each other. Is it a plus sign? I swear I used to be able to do that. Anyway, that's fine. But then here we have another one of these dehydrogenases. And the dehydrogenase is going to release some hydrogen. But what do we notice here? What happens? What else gets released in this step? CO2, which means we're going to go from being a 6-carbon molecule to being a 5-carbon molecule. Does that make sense? So now we go from 1, 2, 3, 4, 5, 6, down to 1, 2, 3, 4, 5. So here, this is a big thing. This is carbon removed. And of course, that's going to be associated with a big release of energy, right? Because we said that catabolic reactions release energy. So what do we notice happens here? We capture energy, and specifically we capture it in NADH, bless you, which is the biggest energy holding molecule we have, right? It holds 3 times as much energy as an ATP. Then something interesting happens, which is we do the exact same thing again. So we used a dehydrogenase to pull off a carbon dioxide, and then what do we use here again? That leaves us with this molecule called alpha-ketoglutarate, a 5-carbon molecule. And we use alpha-ketoglutarate dehydrogenase, which is going to do the exact same thing. What happens? We remove a carbon. So now we've removed all of the carbons that we had, but we still have a bunch of the energy in there because we can still make a lower energy shape. Getting back to oxaloacetate is going to be lower energy. But again, we removed carbon, and again, we captured the energy. Now we get this thing called succinate, but the succinate does an interesting thing. At this stage, succinate gets stuck to an old friend of ours. Who shows up again? Good old coenzyme A, always helping us out. Coenzyme A also likes to stick to succinate, which is this 4-carbon molecule.
But it's more attractive to the.
It works for both. It has 2 jobs. Some of us have 2 jobs, right? Just like we have 2 jobs, coenzyme A has 2 jobs. One of its jobs is to stick to the acetyl group and carry it in here as acetyl coenzyme A. And one of its other jobs is to stick to succinate and form succinyl coenzyme A. And then, what do we have here? The enzyme succinyl-CoA synthetase. Its job is to remove the coenzyme A from it, and as it removes the coenzyme A, it captures a bit of energy. GTP will later go on it'll get processed to ATP later on. When we remove the coenzyme A from the succinyl group, now we call it succinate. And remember, how many carbons should we have now? We took the 4-carbon oxaloacetate, we added 2 carbons to it in the form of our acetyl group. Then here, we removed one of them from isocitrate to form alpha-ketoglutarate, which had 5 carbons. Then we removed another carbon dioxide to make the succinyl group, which has 4 carbons. And now that it's this molecule all by itself, succinate, it should be a 4-carbon sugar. Not a sugar, but a 4-carbon molecule. And now it's going to be all about getting energy out of it. There's no more carbon to be removed. So how do you get more energy out of it? Literally, we're going to change its shape. Does that make sense? We're just going to keep changing the shape so that every time we change its shape, we're changing it into a lower energy shape, a shape with less stress on it. And every time we change it to a shape with less stress on it, it takes less energy to hold the molecule together. That energy has to go somewhere. It's going to get released, and we're going to try to capture it. Does that make sense? So that's what's happening all from here. So succinate is going to get dehydrogenated. We're going to take some energy out of it again to form fumarate. Fumarate is still a 4-carbon sugar, but it's a lower energy 4-carbon sugar. And so because it's a lower energy 4-carbon molecule, what can we do here? We can capture energy. FAD getting reduced to FADH2 is another one of our energy storage molecules. All right, that leaves us with fumarate. Fumarate can have a water added to it to form malate. The enzyme that facilitates that is called fumarase, which sounds like something you have done to an apartment that is infested, but it's not. So fumarase will take fumarate and use it to form malate. And then malate can be dehydrogenated again to form oxaloacetate. Every time we have one of these dehydrogenation reactions, it lets us capture energy, right? But what does that leave us with? This oxaloacetate, or as you see it here, oxalate. This is like the British spelling and pronunciation, and I can never say it. Oxalate acetate. Can't say it. Anyway. But oxaloacetate is how we normally say it over here. And what is that? Well, that happens to be our starting point, which means now this molecule can float around waiting for the next acetylenzyme CoA to come around, and acetyl CoA can add an acetyl group to it, forming citrate, and then it can go through the whole cycle all over again. Does that make sense? Now, we'll talk later on about what I do and don't want you to memorize from this. Boy, if you take a protein enzyme biochemistry course, like in second or third year, they'll want you to know every structure, every enzyme, right? And that's not as hard as it seems, because if you see here, even if we're just tracking how many carbons, it's actually easy to track because when we add the acetyl group, it goes from 4 to 6. It stays 6 until we release a CO2, in which case it's 5, until we release a CO2, at which point it's 4. And then since we don't release any CO2s, is that going to change? No. All we're doing is changing shape. Does that make sense? And when I took that protein and enzyme biochemistry course, the way I used to study was actually just build it. I would build oxaloacetate with my model kit. I would build an acetyl group with my model kit. I'd put them together to make citrate. And then I would change them to make isocitrate. Then I'd change them, I'd take off a CO2, and I'd build an alpha-ketoglutarate. And I'd just keep doing that. And you see, like, okay, there's a logic to this. It makes sense. It's not just random things. Because it's a process. Yeah. 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 And I'm going to show you that, actually. And because MolView is really slow today, it's going to take a frustratingly long time to show you. But that's okay.
You should try to beat it and build a model of it.
I don't think I could do that. So let's actually start with succinate. Oh, great. Great. Here we are. So succinate. And again, how many carbons should be in succinate? 4. So look here, 1, 2, 3, 4. And if we look, sorry? Faster than that? That was pretty quick.
Oh, I thought she meant like just build it.
Oh no, I could build it, but not faster than that loaded. So if you look here, succinate is just another word for 1, 2, 3, 4. So it's a 4-carbon molecule. If you've taken organic chemistry, you'll know that is a butyl group. Good. And this is dibutanoic acid, right? It's just got an acid group on each end, right? We recognize our carboxyl group there. It's not showing the hydrogen because it's dissolved in water. So now, from succinate, when it gets turned into fumarate, you can see that the difference is it has lost hydrogens. Do you see that? And it's formed a double bond here. If you've taken 4U Chem, you know that we call that a subtraction reaction, right? Like we've removed those and we've formed a double bond. So that's why this is called a dehydrogenase, because what got released? Hydrogens. Then, from fumarate, we actually are going to add back in those hydrogens and an oxygen while we're at it to form malate. And again, where does it get the water? These cells are full of water, right? Lots of water available. And we get malate, it's going to be back to being a 4-carbon structure. Now, it actually looks like the succinate, but it has an additional hydroxide group. Do we see that? That's okay. And then that malate is going to get dehydrogenated again. So we're actually going to take hydrogens off again, forming a double bond again. And this time we're going to form oxaloacetate. And there it is, right? So we lost hydrogens again. This time, though, not forming a double bond between the carbons, but actually forming double bonds between the carbon and the oxygens. But that's oxaloacetate there, and that is our starting point. Does that make sense? So if we watch it again, this is the starting point. When we throw an acetyl group on it, we're going to get citrate. If you look at citrate, there it is, 6 carbons, right? 1, 2, 3, 4, 5, 6. And that's that oxaloacetate, but with the acetyl group thrown on here. Can we kind of see that? Then, from citrate, we're going to get isocitrate, which is just that, but transformed. It's an isomer of citrate. Maybe that's why they call it isocitrate. Then it's going to get dehydrogenated, which means we're going to do what? We're going to take hydrogens off. So when we dehydrogenate isocitrate, we're also going to remove a carbon, forming alpha-ketoglutarate. Alpha-ketoglutarate, instead of having 6 carbons, has 1, 2, 3, 4, 5. But you can see it's also lost a bunch of hydrogens, right? It's now got these double bonds to oxygen. That's why they call this a dehydrogenase. And along the way, NAD formed NADH. which stores a bunch of that energy, right? The alpha ketoglutarate is going to get dehydrogenated again. Where's that going to happen? Well, there's not much hydrogen left, right? So do we think maybe we're going to get a double bond between carbons? Let's see what succinate looks like. I'm going to skip the succinyl coenzyme A, but if you see there, we've got double bonds now, and there we go. And then from succinate, because you're in water, so there'd be a negative charge there. Does that make sense? It would have an H on it. If you were to take this and solidify it and crystallize it, it would have a hydrogen there. But when it's dissolved in solution, because what group is that? This whole group is a carboxyl, and the whole point of a carboxyl is that hydrogen can be released. That is the acidic hydrogen. So when it's dissolved in water, we don't have a hydrogen there because it's acting as an acid. So if you were to look up the crystal structure of this as a solid, you'd see it with the hydrogen there. Does that make sense? Cool. And then, we've already done this, but just to show again, fumarate gets processed into malate by the addition of a water. So we actually put hydrogens back in place. And malate, did I skip fumarate? No, I did fumarate. And then we translate that back into oxaloacetate. Yeah, it's funny, they don't even have that stupid spelling there, oxaloacetate. And there it is. How's that feel? Not too bad? So, what is the point of all this? The point of all this is that I don't want to use the language I used here. Oh, sorry, I did want to write a couple sentences here. I forgot that. In this space here, we're not going to write everything out, but in Krebs cycle, I should say the Krebs cycle, because again, it's not possessive. It'd be like calling it the Orlando cycle, as opposed to Orlando's cycle. We put the in front. It takes the definite article. So in the Krebs cycle, an acetate group, which is this C bound to C, making a double bond to O, making a double bond to OH, is added to the molecule oxaloacetate to begin the process. The acetyl group is the source of all the energy that gets extracted. Is everyone comfortable with me doing E as energy, by the way? It occurs to me that I've just been doing that, and I never actually checked in that everyone understood what I was doing, but everyone's cool with that? Okay. So, in other words, as such, when all the energy is extracted, we're left with oxaloacetate again. And that can do the whole process all over again. Does that make sense? So again, why even bother with oxaloacetate? Why not just work with an acetyl group all the way through? The answer is that it's easier to work with bigger molecules. It's easier to make an enzyme that is specific for a big molecule than it is to make one specific for small molecules. So, by attaching the acetyl group to oxaloacetate to form citrate, we are working with bigger molecules and the cell can more efficiently process them. but it's still cyclical, which means at the end you get an oxaloacetate back that you can do the whole thing over again. I gave you a couple other graphics of this, which I want to look at just before we go. Tomorrow, tomorrow we're going to do different stuff. But what's the purpose of all this? Purpose of all this is what I wrote in that space above. Whoops. I should read my note more carefully. Sorry about that. If you look here, this is the same version, and it gives you the structures. I like this one too. So we can see here that we take our pyruvate, and from our pyruvate, we release a carbon dioxide, and we get our acetyl group, which will then stick to coenzyme A, which it shows as a little rectangle, but is actually much, much, much bigger. And then the acetyl coenzyme A will react with oxaloacetate, and the enzyme citrate synthase will use that to form the 6-carbon citrate. And then aconitase will form isocitrate. That's just an isomer of the above, at which point isocitrate dehydrogenase will facilitate removing hydrogen, removing a CO2, taking some energy and storing it in NADH, and leaving us with the molecule alpha-ketoglutarate, where alpha-ketoglutarate dehydrogenase will do the exact same thing and now form succinyl-CoA, where we have this succinyl group stuck to coenzyme A again, at which point succinyl coenzyme A synthetase will remove the coenzyme A, forming succinate. That actually releases some energy in the form of a new molecule we haven't seen yet called GTP, guanosine triphosphate, which is not that dissimilar from adenosine triphosphate. Succinate dehydrogenase can be used to form fumarate. Fumarate can receive a water to form malate, and malate can be dehydrogenated again to form oxaloacetate, which is our starting point, and that releases yet more energy in the form of NADH. By the way, if you look down here, they show this step as quinone forming quinone H2. That is what we show as FAD forming FADH2. It's just two ways of writing the same thing. And this is another energy storage molecule. That's a lot, yeah? It took a bunch of scientists a decade and like a famously chaotic lab experience to do this. I read a great book once about the laboratory experiments that led to the understanding of Krebs cycle. It was like wild and crazy stuff. They were working with like extreme high voltages. People were like badly electrocuted and hurt. Like it was like, oh yeah, it was like a serious Wild West show to figure this stuff out. Because again, it's not like we can stick a little video camera in the cell and watch this stuff happen. We had to isolate components from inside the cell and figure out what was going on. This is a major, major, major discovery that took some of the brightest minds in the world a long time to figure out. So if you feel a little confused by it after one day, yeah, sure, like that's pretty reasonable, right? But listen, look at me. Talk your way through this stuff, all right? Talk your way through it. Tomorrow we're going to have a little quiz. It won't include any of today's stuff, but up to glycolysis in this unit will be fair game, okay? So reread your notes. What I am going to leave you with is a little textbook section to read. Your challenge today is to read up to the part on Krebs cycle. There's practice questions in here you can do, but I'll tell you right now, all of my quiz questions come from the practice questions in here. It's not a bad idea to take a look at them. Is that fair? All right, grab one of those. Thank you, guys. Yeah, it's just like a whole chapter of the textbook. I'm trying to figure out how to buy textbooks right now without spending my entire science budget. Do you know that my entire science budget is enough to buy 27 textbooks? So that's challenging, right? Because I also have to buy gloves and chemicals and... Sorry? That's true. So there you go. Now, the flip side to this is this will end up costing us a lot more than textbooks because paper and photocopying ain't cheap. But penny wise, pound foolish, as they say. Do you know that expression? No? Am I outing myself as an old man by saying penny wise and pound foolish? Simpson, how you doing, dude? Yeah, scale of 1 to 10. 3? Oh, no.